peeling apart at high temperatures. Virtually all hyperthermophiles contain lipids constructed upon the diphentyl tetraether model. For the structure of such lipids, diphenyl tetraether lipids are naturally heat resistant because the covalent bond between phenyl units forms a lipid monolayer cytoplasmic membrane instead of the normal lipid biolayer. And figure, this structure resists the tendency of heat to pull apart a lipid biolayer constructed of fatty acids. Besides the stability of macromolecules, the thermal lability of monomers is important in governing the upper temperature limits for growth of hyperthermophilic bacteria. No matter how stable the macromolecules, life is impossible if the basic building blocks themselves are unstable. The thermal stability of small molecules rather than macromolecules may therefore dictate the upper temperature for life, and even at temperatures as low as 120 degrees Celsius, some important small molecules are destroyed at significant rates. For example, two key molecules in energy metabolism, ATP and NAD, hydrolyze quite rapidly at these temperatures. Their half-life is less than 30 minutes at 120 degrees Celsius and shortens dramatically at temperatures above this. With this in mind, what is the upper temperature for life? The limits to microbial existence, because life as we know it depends on lipids, water's microbial habitats hotter than 100 degrees Celsius are only found in environments under pressure, such as the seafloor. Indeed, all hypothermophilic archaea capable of growth over 100 degrees Celsius seem to be restricted to these superheated environments. An answer to this question has been sought from the study of deep-sea hydrothermal vent black smokers as models for superheated natural environments. Smokers emit hydrothermal fluid at 250 to 350 degrees Celsius and form upright metabolic structures called chimneys from metal sulfides in the fluid. Although hyperthermal phials have been isolated from smoker chimney walls, which show a gradient in temperature from 250 degrees Celsius inside to 202 degrees Celsius outside, studies of the 250 degrees Celsius water itself show it to be sterile. This is consistent with laboratory measurements that have clearly shown that important biomolecules would be instantly destroyed at such a high temperatures. Laboratory experiments suggest that living processes could be maintained at temperatures as high as 140 to 150 degrees Celsius, but that above this temperature organisms would probably not be able to overcome the heat lability, the essential biomolecules of life. For example, if organisms exist that are capable of growth at temperatures even as low as 150 degrees Celsius, their energy economy would almost certainly have to be based on something other than ATP, the energy currency of all other forms of life, because ATP is unstable at this temperature. Thus, if superhydrothermophiles capable of growth at or above 150 degrees Celsius are ever discovered, they will likely demonstrate some fundamental new principles in biology and biochemistry. Hyperthermophilic archaea and microbial evolution. An interesting question concerning the archaea, why so many of them inhabit extreme environments, although molecular probing of non-extreme environments like soil and water indicate that archaea are all around us. A common theme running through cultured representatives of the archaea is adaptation to environmental extremes. As we have seen in this chapter, various species growing above the boiling point or at extremes of pH or salt are known. Is this a mere coincidence or a reflection of Earth's evolutionary history? Extreme environments of various types existed on early Earth as they do today, and it is within such environments that life may first have flourished. At the time that cellular life first evolved, it is almost certain that Earth was far hotter than it is today, and the hypothermophiles in particular stand out as the best extant relatives of Earth, early life forms. What does the phylogenetic tree of Archaea tell us about these hypothermophiles? Molecular sequencing of rRNA suggests that many archaea evolved at slower rates than most bacteria or eukarya. This is especially true at the hypothermal archaea, both eukaryotes and crinoarchaeotes. Much the same can be said about the hypothermophilic bacteria such as Thermodiga and Aquaflex. This conclusion emerges from the inspection of the evolutionary tree lines, and the hyperthermophiles are typically rather short and branch near the root. It is not known why heat-loving archaea have such slow evolutionary clocks, but one hypothesis is that this is a consequence of their inhabiting such extreme environments. Organisms living at very high temperatures may be under unusually strong evolutionary pressure to maintain those genes that specify phenotypic characteristic critical to life. That is, beyond a certain point, survival of hypothermophiles may not be furthered by additional genetic change. If this hypothesis is correct, then hyperthermophilic archaea are indeed living relics of the earliest life forms, and their continued study should reveal important principles of early Earth biology. The slow clock idea can be further explored in the phylum of Archaeota, which are shown in the phylogenetic tree. As the least derived of all archaea, unfortunately we know very little about this group. Representatives of the Archaeota have thus far been identified only from the single hot spring, Yellowstone National Park, and the group is not recognized taxonomically, thus the use of quotes around the name. However, mixed laboratory cultures of Archaeota that clearly indicate that they are hyperthermophiles, as would be expected of prokaryotes close to the root of a tree of archaea. Further progress on this interesting group is anticipated and may yield secrets about the early Earth and early life forms. Hydrogen is primitive energy source. Before we leave this chapter, we wish to point out how often H2 metabolism enters the metabolic picture. Many archaea grow anaerobically with H2 as an electron donor and one or more of the electron acceptors. S to the power of 0, NO3 negative, and Fe to the power of 3 plus. H2 metabolism is likely a physiological relic of ancient metabolism schemes. It gives an example of one that first evolved in primitive organisms because of early ready availability of these organic compounds. In their primordial environment, the diversity of H2 oxidizing hyperthermophilic archaea and bacteria. Today, and the fact that so many mesophilic bacteria, distant relatives of the hyperthermophilic H2 oxidizers, also use H2, attest to the evolutionary of H2 oxidation as a metabolic success story, and the use of H2 by hyperthermophiles and various physiological groups of prokaryotes living deep in the earth are prime examples of the metabolic strategy. Both hydrothermal vents, the habitat of many of the most extreme hyperthermophiles, and earth, hot deep subsurface, have been suggested as the types of environments where life could have first arisen. These environments contain an abundance of inorganic electron donors and acceptors and are free from exposure to ultraviolet radiation. It is thus possible that the hyperthermophiles discussed in this chapter are truly relics of the early Earth. Well, this is the end of chapter 13, and good night.